Hello everyone, welcome to Easy Med. Today's our topic of discussion is iron metabolism. It contains absorption, transport and storage of iron. Absorption of iron has three steps: internalization, mucosal block and transport from enterocyte to plasma. Internalization is the process of absorption of iron from alimentary canal, that is duodenum and proximal jejunum, to enterocytes, which are cell of interstitial lining. Food iron may be heme iron, that is Fe2+, which is mainly obtained from animal source, and non-heme iron, that is Fe3+. These are mainly obtained from plant source, but only the heme form of iron can be absorbed by the body. So the non-heme form of iron is converted into heme form, that is Fe2+, by the action of enzyme ferroreductase, and is transported into enterocyte via divalent metal transporter 1. In absorption of iron, the next step is mucosal block. Duodenal cells synthesize a protein called apoferritin. Apoferritin traps the internalized iron and forms mucosal ferritin, and some of the iron is left unbound or free. Only this unbound iron can go to circulation, whereas the bound iron gets trapped and lost in feces by shedding of cells. Thus, amount of iron absorbed is inversely proportional to the synthesis of apoferritin. That is why during iron deficiency or increase in iron requirement, synthesis of apoferritin is inhibited, and when body has enough iron stores, synthesis of apoferritin is increased. This mechanism is called mucosal block or ferritin cotton. The next step is transport of iron from enterocyte to plasma. Iron is transported from basolateral membrane of enterocyte by ferroportin. In plasma, Fe2+ is oxidized into Fe3+ by hephaestin and ceruloplasmin. Now, moving on to transport of iron. From plasma, iron goes to circulation. In circulation, transferrin is present, which is synthesized by liver. In circulation, the absorbed iron is trapped by transferrin and carries iron to the site of utilization and site of storage. Target cells like RBC, bone marrow, liver cells and spleen, they express transferrin receptor like TGFRN1 and TGFRN2. Transferrin along with iron binds to this receptor, and then the iron is internalized by this cell by endocytosis, and free transferrin, that is apotransferrin, it is released into circulation, which is again used for transport of other iron. Around 75% of the iron goes to bone marrow, where it is utilized for erythropoiesis, and 25% of iron goes to the site of storage. Talking about the storage of iron, iron is stored in liver, spleen, bone marrow and skeletal muscles in the form of ferritin and hemosiderin. In liver, most ferritin is stored within the parenchymal cells and other tissues such as spleen, and in bone marrow, it is mainly stored in macrophages. Let's talk about hepcidin. Hepcidin is a master iron regulator synthesized by liver in response to increase in intrahepatic iron level. It acts on ferroportin in basolateral membrane of enterocyte and decreases transport of iron from enterocyte to plasma, causing decrease in plasma iron level. So when iron stores are reduced, synthesis of hepcidin is inhibited. That was all about the iron metabolism. Hope the video was useful, and thanks for watching.